Here's an exercise for you. Write out 50,000 nucleotides for me, A, T, C's, and G's. Just 50,000 A, T, C's, and G's in whatever order you would like. Just start writing them down. A, T, A, T, 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 A, G, C, A, T, C, G, 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 C, G. Give those 50,000 base pairs to me. And I will synthesize that 50,000 base pair strand of DNA. Now, what if I were to take those 50,000 base pairs, that random code you just gave me, and I were to go into your genome and splice open one of your chromosomes and stick that 50,000 base pairs in there? What do you think would happen to your genome? Would it damage what's already there? Would it add something new? Is it new information being added to your genome? How would your cells react to this extra 50,000 base pairs? Could there possibly be information contained in that 50,000 base pairs, even though you randomly wrote out those base pairs yourself? You are a designer of sorts. You designed those 50,000 base pairs, but if you had no sense of what you were making when you did A, T, A, C, G, A, T, G, C, is it possible that it could make something? Or do you truly believe that what you've done is you simply added some junk to your DNA, to your genome? It's worthless pieces of information that you've just transposed or inserted into your genome. We're here to explore that question. And I'm going to do that by showing you some bioinformatics. We're going to generate a 50,000, actually we're going to start with a 5,000 base pair DNA sequence. We might do a 50,000 or even 100,000 base pair sequence, randomly generate that. And then we're going to ask ourselves, what is that 5,000 or 100,000 base pair fragment capable of doing? How would your cell react? How would any organism's cell react to that extra DNA that's been added to its genome? We've got that coming up. Okay, before we dig in, I have to explain kind of the style of what we're going to do here. This is an experiment for me to see whether I can handle doing something maybe more live-ish than I have, right? I usually do recorded stuff and then I splice all my faux pas out, right? before I allow you to see what I've done. But what I've done now is I have a whole bunch of websites. I have some ideas right here in my head about what I want to cover and do. And we're just going to get started and we're going to explore this question. If I were to generate some random sequence and insert it into a genome, what might happen to it? What are the possible consequences of that? Now, I have a reason for doing this exercise. It's something I've been meaning to do for quite a while. I wanted to actually explore this myself through some bioinformatics tools. And I've played around with it a little bit, but I'm going to be generating all new stuff for you that I've not done before. And so whatever happens is what's going to happen. Okay, back to the reason. Why am I doing this? What was my initial impetus for thinking through this little question? And if you've followed me in the past, or maybe seen me comment on other other YouTube channels, I've asked this question before, and it's in a particular context. And the context has to do with the ENCODE project, or creationists or anti-evolutionists who will say that there really isn't any junk DNA, right? There isn't any useless DNA, like all DNA has a purpose. And one of the reasons they say that is because it's often pointed out that evolutionary biology evolutionary biology explains the fact that our genomes are absolutely massive, or most organisms' genomes are very, very large, and they're only using, now it's tricky to define the word using, but they're only using a portion of that genome, and the rest of it is we consider kind of leftover remnants of past genes, past programs that organisms have used and have discarded, but they're just still being copied along the way. But creationists typically will say, like, everything in the genome must have actual usefulness. Otherwise, why would it be there? Like, why would a creator have placed hundreds of thousands of base pairs in a genome? They're just random garble that don't mean anything, that they don't make anything. They have no actual purpose, sense of function in the genome. And so they'll point to, oh, look at these new studies showing that, we use much more than just a couple percent of our DNA for coding sequences, but all the rest of the genome is actually being utilized or has some function. And they point to a project called the ENCODE project. And the ENCODE project, what's involved in the ENCODE project is, oh, you know what? Why am I talking about this? Why am I stumbling over it? I need to just go to the article and show you. There was a reason why I got my tabs ready. And I've already forgotten what that was. I could go to a lot of different young Earth creationist or intelligent designist sites to provide information about the ENCODE project or the complaints that, or the arguments they have about whether the genome is functioning or not, or how much the genome is functioning, or what is this thing called junk DNA, and really junk DNA doesn't exist. But I'll just take you to one, because we don't need to spend a lot of time talking about that when I'm really going to just be exploring this other question, or I can actually show you what DNA does or can't do. And I think you'll get the sense for why I am doing what I'm doing here. So let's see, decoding the debris. This is a article by Georgia Purdom, and I picked this one out because she is a molecular biologist, all right, molecular geneticist. That's what her PhD is in from Ohio State University. And she works at Answers in Genesis, which is a young Earth creationist ministry. For decades, evolutionary scientists have ignored junk DNA that supposedly littered our genetic sequence, right? That there's this useless stuff in our genome. And now they've finally realized their mistake. Now, what's she referring to back in 2014? She's referring to the fact, she tells this story about where the term junk DNA came from. And a lot of this is a big oversimplification, but I'm not concerned about that right now. We just want to get down to this. In 2003, the ENCODE project, that's an acronym for the Encyclopedia of DNA Elements, was launched to evaluate the role of non-coding DNA. Now, non-coding DNA would be DNA that doesn't code specifically for proteins. You might have learned that typically when we think of genes are segments of genetic code, ATCs and Gs, some sequence of code that is transcribed into a message called the messenger RNA. And that messenger RNA is then read or translated into a series of amino acids in a particular order. That order is governed by the order of the ATCs and Gs. And that order of amino acids then folds up into a structure we call a protein. And so the proteins, the instruction set of our genome is an instruction set for how to make a diverse set of proteins. And proteins make structures, like my hair, the small hairs back here. And they make enzymes, and enzymes govern chemical reactions. So therefore, this is the code. Our code codes for enzymes which govern chemical reactions and therefore drive all the different chemical reactions that put things together and take things apart to make our physical being. And it's been known for a long time that really only a very small portion of the genome is collectively all of your DNA code, all of your 3.3 billion ATCs and Gs on one copy of your genome. Only a very small percentage, at about 2%, is actually utilized for coding your proteins and making your proteins. So the question is always, had been, what does the other 98% do? And clearly some of that has other functions, because as I'm going to show you, there's many things that control the how you turn on genes, how you regulate genes, you enhance genes, you suppress genes. All those types of regulatory sequences are not actually, in many cases, not proteins themselves, but other transcripts of your portions of codes that then are influencing other things. All right. Now, that's been known for a long time, But nonetheless, the ENCODE project came along and it was trying to make an encyclopedia of all the other different elements of your DNA code. What out of the 3 billion base pairs, what's it all doing, or is it doing anything at all? And what they concluded in 2012 in the very first paper was they estimated that about 80% of the genome is functional in some sense. Now, I'm going to read her wording, Georgia Purdam's wording. And Georgia Purdam's very much has an agenda here. She is very much trying to use the secular literature to make the point that she would like to make, which is that, look, most of your genome has function, right? It has purpose, right? And this is against the typical evolutionary model in which over millions of years, some portions of the genome have lost their function and they're just sort of hangers on. But no, in the creation model, everything must have had a purpose, since why make more than you needed to make an organism? More genetic information than you needed to make an organism. So therefore, the expectation is that all portions of a genetic, all A, T's, and C's and G's serve some functional role or purpose. Okay, so the ENCODE project first estimated to be about 80%. Now, I have to read this carefully. Since in a specific biochemical activity, in the sense that it has specific biochemical activity. I just said that twice, and I said it for a reason, because we're going to look to see what is really meant by biochemical activity. Right? And she does suggest that this is a broad general term that has stirred debate. Yeah, it has stirred debate. I will get into that. Much of the function is believed to be regulatory. Okay, yes, some of that function is regulatory. In the simplest terms, non-protein coding parts of the genome, formerly called junk, are telling the genes when to produce their products or proteins as a whole, and a lot more. She's making it sound in this particular article as if most of the 3 billion base pairs have some regulatory properties, and that they have biochemical activity, like they're being utilized for some purpose in terms of generating how organisms function. This is saying far more, actually, than the ENCODE project says. But that will become evident as we go through our little experiment, and we actually kind of test this idea as we will actually construct and make random DNA. And then we'll ask ourselves, this is really what we're going to do. We're going to ask ourselves, how much of that random DNA would have biochemical activity? How much of that random DNA would make proteins? How much of that random DNA that any one of you could generate and make would itself be labeled as having function, given her definition here of function? All right, that's what we got to get into. That's really the bottom line of where we're going with all this. Okay, so this is my inspiration, because I hear this all the time on blogs and YouTube channels and other places. Oh, look, the whole, most of the genome has function. And the other thing we hear all the time is from young Earth creationists is no new information can ever be added to an organism. Right? Evolution can't add new information. It might be able to tweak and play around with the information that's already there by having small mutations and maybe make some new variants of a particular code, a particular sequence. But we're going to ask the question, if you add a chunk of new DNA to an organism, is that new information, and can it actually make new functions for that particular organism? How are we going to do that? Well, let's do it. Let's get started. All right, let's try out full screen. Remember, you're watching an experiment in progress as I learn how to show things in different ways in videos. So we're at the bioinformatics.org sequence manipulation suite, and it's a whole series of programs. All these links down the side are to different tools, and we're going to scroll down here to random sequences. So we could mutate DNA, we can mutate proteins, we could make random coding DNA. All that would mean it would have a start codon, stop codon, the place that tells a protein to start reading and a place for the translation apparatus to tell it to stop reading, and everything in between is the protein. We just want to make plain old random sequences. So I click on random sequence. Random DNA sequence generates a random sequence in the length you specify. All right, so into the length of sequence, max accepted value is, well, I'm sorry, but the biggest piece of sequence we can make is 10 million base pairs here. Okay, so we're going to start out simply. We're going to start out with 5,000. I want to generate 5,000 base pairs. And I only want to generate one of these. Let's hit submit, and boom, that didn't take very long. There's our 1 to 5,000 base pairs. So here we go. A, T, A, C. What we got? C, T, T, A, T, A, C, T, A, T, 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 G, T, C, G, G, T, G, 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 C, G, C, T, T, A, T, C, T. You got the idea? 50,000 base pairs. I mean, we could have done that the hard way. Could have had you, one of us could have written this out from our head, although we tend to make patterns as we do things. This is a little less pattern, so we'll say a little more random. All right, so we've got the sequence. Now I'm going to copy that sequence. All right, so we've got our 5,000 base pairs. We want to look now, and we want to ask ourselves now. Oh, yeah, let me say what I could do. All right, with that sequence. What we could do with that sequence is it's possible to synthesize large pieces of DNA. It's hard to synthesize a really large fragment like that, but 5,000 base pairs is doable, right? We could just take that sequence. I could send it to a company. I could just tell them, send me in a tube strands of DNA that are 5,000 base pairs long and have exactly the sequence I just told you to make for me. So we'd have, in my lab, I'd have a 5,000 base pair string of DNA with that particular code in it. And so I couldn't ethically do this. But what if we were to say that now I'm going to take that and I am going to, let's do it in cell culture. All right, we got a whole bunch of cells. And we're going to do some, we're going to do some DNA manipulation, right? We're going to take the DNA strands I've got, and we're going to insert them into the genomes of those cells, or more particularly one cell. And then we'll try to grow that cell into, like, say, a couple 100,000 cells. And so if you copy, you open up a chromosome, splice in that 5,000 base pairs, and then you have it in that cell and have the cell copy itself. It's going to copy that 5,000 base pairs. And then you're going to have a whole bunch of cells that all have that 5,000 base pairs in it. And then we can do some kinds of interesting things to it. Like we could extract all the messenger RNAs from those cells. The messenger RNAs represent all the messages that the cells are making from their genome. And we could sequence all those messages. And we could see, are some of those messages actually coming from our new 5,000 base pair strand of DNA? Like maybe there's actual information in the 5,000 base pairs of random sequence we put in there. We might actually see it being generated in those cells. But we'll come back to that. First, we just want to ask, before we ask what the cells are going to do with it, let's ask ourselves, is there any information in that random 5,000 base pairs? And again, I've done this already, but I didn't do it with this 5,000 base pairs because this just generated a brand new 5,000 base pairs for us. So I'm going to go over here and I'm going to go over to ORF Finder. All right, now ORF Finder, ORF stands for Open Reading Frame. All right, now open reading frames are what open reading frame is what we refer to as where potentially there is a protein encoded in a sequence. So if I were to sequence your genome and I got like 3 billion base pairs of your genome and I didn't know where any of your genes were, I could start by using a program like this, Open Reading Frame Finder. And it would go through and it would look for everywhere along your DNA strand, both on the one strand and the other strand. Remember, you're double-stranded, and some of your genes are actually coding in one direction and some are coding in the other direction. So it would scan both directions and would ask, where is there an ATG going forward down your forward strand, and where do you have an ATG going the other direction? ATG is the relatively universal what's called start codon. That's a position where your translation apparatus that's going to read a protein will look for an ATG and it'll say, oh, I see an ATG. ATG is methionine, the very first amino acid of almost all proteins. And so that effectively is the place where you start making a protein. And so if you just scan your entire genome, you can find every start position that you have, or we should call it potential start position. And then it will also look for downstream. It'll say ATG, and then it will start looking. It'll start scanning down the sequence. And what it's looking for is it's looking every 3 bases, because every 3 bases is what makes an amino acid. That's what your code does. Your code is ATC, TTA, GGG, GCC, ATG, right? And those 3 individual bases correspond to different amino acids, which make the proteins. So it's going to look every 3 base pairs. And what it's looking for, what this finder is looking for, is it's looking for a 3 base pair sequence that is a stop codon, the signal for the translation apparatus, the machine, to stop reading the code. So you had a start position, and now you're going to have an end position, a stop position. And that's, we'll say, in our case, it's TAA. Right? There's actually 3 stop codons, but we'll pick a specific one, TAA. And so it's going to look for the start, and it's going to look for a stop, and it's going to say, oh, I found a possible position where there is a protein coding gene in this sequence that you just gave me. Oh yeah, that's right. We got to give it its sequence. So I'm going to paste it in there. And I just pasted it in there, random sequence results. I'm going to take that out. So we have random sequence 1 consisting of 5,000 base pairs. There's our 5,000 base pairs. There they all are. That actually scanned up pretty quick there. I'm going to say ORFs can begin, and we're really looking for ATG because that's the most common start codon. We're trying to keep this simple here. There are exceptions to almost every single rule in genetics. There's an exception. We're going to search for ORFs in reading frame 1, 2, and 3. That just means it can start on base pair 1 or base pair 2 or base pair 3 because we don't know where the reading frame is. And we're on the direct strand. We're going to look down the forward strand first. And let's return any open reading frame that is 15 codons or longer. Each codon is 3 base pairs. So anyone that's 45 base pairs or longer, so that would make a code that is 15 amino acids long, or a protein that's 15 amino acids in size. You ready? Let's do it. Submit. Wow, okay, there's our result already. We're working on a really fast computer here. Not mine, mainframe computer somewhere. Results of 5,000. So we have ORF number 1 in reading frame 1, and the direct strand extends from 256 to 384. So this sequence right here, you see how it starts with an ATG? And at the end it has TAA. So in our randomly generated 5,000 base pairs, we happen to randomly generate a sequence that has a start codon and a stop codon. And in between it has, I can't count that up, 256, it has 128 base pairs. And it provided us its translation. So this would be the protein right here. Starts with M, which is methionine, and then ends with this asterisk, that is the stop codon. All right, so that particular sequence in our random sequence we generated would correspond to this particular very, very small protein. Let's look down here. We've got a third one. This one's a little bigger, so this is a decent sized amino protein. We've got 2,824 base pair 2,824 to base pair number 3,126. So now we've got ourselves we have 302 base pairs. All right, 302 base pairs, which is divided by 3 is about 100 amino acids. That's 100 amino acid sequence that could be generated by our random sequence generator. And here, this would be the protein. And so each one of these letters corresponds to a different amino acid. The amino acids would be all hooked together, and then they would fold into some kind of structure. Now, of course, we just made a random sequence of amino acids. A random sequence of amino acids could still be constructed by the cell, but when it folds, it may not fold in any sort of particular pattern, but it's going to fold into something. All right, it's going to make some crazy structure. And after it makes that crazy structure, it's going to be in the cell potentially as this particular protein. And then is it going to have function is the question. And I would say probably not, right? There's a very, very, very high chance that this little glob of protein will have nothing to do with any of the activities of the cell. But it could by chance be very similar in some section of it and bind to another protein. Right? Proteins bind to other proteins. There's lots of proteins that form large complexes of proteins. And if this protein binds to another protein, or binds to a piece of RNA, or binds to, well, could bind even to DNA. There's lots of proteins that bind to DNA, and DNA binding proteins, what do? They either turn on or turn off transcription, right? The process of turning genes on and turning genes off. They regulate genes. So if this protein by chance has a particular sequence, and it might just be a series of 7 or 8 amino acids in a row that is very similar to another DNA binding protein, it might actually bind to something in the cell. And if it binds to something in the cell, it likely will have a small functional effect on the cell itself. All right, let's keep on going down here. We've got 4, 5, 6, open reading 6, open reading 7, 9, 10. And then we have on reading frame 3, we've got multiple different proteins up to 12 there. So we've got 12 there, 11 there, and 6. Oh, sorry, 5. I think I got to do some quick addition here. 5, 11 is 16. 16 plus 12, 28. There are 28 open reading frames that are of 15 amino acids or larger that are in our random 5,000 base pairs we generated. 28. And we just did the forward strand. We didn't do the reverse strand. Oh, let's do it real quick. Because I could say, let's do the reverse strand. That means it's looking down the other strand, the opposite direction, which has the opposite base pairs. And so you have the ability to scan that as well. We have genes on both sides of our DNA strands, our double strands. Let's do that really quick. And what we get there is we've got 12. We've got 8, 9, 10. We have 11 plus 7 is 18 plus 12. We have 30 on the other side. So we have 58 total. 58 possible locations where there's a start codon and a stop codon. And there's a sequence in between that can be read into a protein. Now, but you have to ask yourself now, okay, will it be read into a protein? Just because it can be read into a protein. In other words, the information is here that if the translation apparatus of the cell is able to see this particular sequence and see that start codon and see that stop codon, it very likely can read that code and it will spit out a protein. Now remember what Georgia Purdom said about what makes, in code found, what did they found? They found that 80% of the genome has biochemical activity, right? Biochemical activity would mean that the code can be expressed, like turned into transcripts, messenger RNA, I'll show you that in a moment, or turned into proteins. But those proteins don't necessarily do anything. In other words, they don't necessarily have function. Biochemical activity is very different than actual functions, right? Lots of things can have activity, but the activity doesn't amount to any actual functional thing that you would consider at all important for the life of that particular organism. Okay, we're going to need to use a different tool because we have to ask a different question now. We have to ask, okay, before an organism can make a protein, they have to transcribe it. They have to get the information out of the genome in a format that can be read by the translator. All right, so this, all I've shown you now is there is something there that can be translated. But before it can be translated, it has to be transcribed. It has to be brought to the appropriate location in the cell. And there's a lot involved in that. Let's talk about what's involved. So let's get rid of that. Let's go here. Okay, now this is going to look pretty complicated, but in order for, you've got a string of DNA. Oh, you know what I need? Let's go. All right, we have your DNA, and we've just inserted this 5,000 base pairs inside of somewhere in your DNA. All right, and in that 5,000 base pairs, what we're saying, what I've been saying, is that we just located 30 different locations. You can think of these as little boxes. All right, inside of these little boxes, these are little boxes that have, if we expand one of these out, we've got an A, T, I wish I had a better, I don't have a pen, so I'm doing this with my mouse. ATG and a TAA at the end. Okay, a TAA at the end. Oh, that's horrible. And in between, there's a set of sequence, right? It's C, C, C. I'm just doing C's because I can draw them easier. In between. And we're saying that, so this is the start and this is the stop. And so potentially there is a gene right there, because a gene is something that codes for a protein. Now, whether you want to say the protein has function or not, a gene that codes for a protein, a sequence that codes for a protein, we can call a gene. Right? It might be a non-functional gene, but it's a gene nonetheless. And so here we've got this sequence in your DNA code. But in order to make this particular code and translate it into a series of amino acids, methionine, followed by, I don't know, cytosine, followed by proline, followed by arginine, all right, and then stop at the end, TAA. Right? In order to do this, you first have to get the code out of your genome, right? Out of your nucleus. And in order to do that, you have to copy this piece of code. So you have to copy this code, and you have to bring it out of the nucleus and bring it to the location where it's going to be read. It's called the ribosome. That's the machine that reads your DNA and actually looks at the ATG and goes like, I know what to do with ATG. I need to make it into methionine. All right. How do you copy your code? Like, how do you know that that along your DNA code, because you have a gene down here and you've got a gene down here on the other side of where we spliced your 55,000 base pairs in, right? And now you've got this little segment. It has a start codon and a stop codon, but you can't read that code directly off of into a protein directly off your DNA. You have to take a little copy of it out of your genome, where you're protecting it inside your nucleus. That's what your information is. It's like your hard drive on your computer. You've got your programs there, right? And then when you want to go use your program, what do? You go in and you find where the program information is, and you make a copy of it, and you bring it out and you put it in your random access memory, your RAM. And then you execute it. All right. You can think of that as your genome is your hard drive. Then you take a copy of portion of your genome. You have to go find sections that you need, and then you make copies of it, and then you bring it out to the ribosome, and your ribosome is your CPU. And it says, oh, I know what 0101100100100 is. I can read those zeros and ones, and I can turn it into pixels of color, whatever you're telling, whatever that program is saying to do. So we need to copy this particular code. I'll find out where I'm at here. We need to copy this particular code and get it out of the nucleus, out of the double-stranded DNA. I got my double-stranded DNA molecule. Here, maybe I should bring it over here. Right? So I got my double-stranded DNA molecule, and here are the pieces of code inside of here, right? And we made our 5,000 base pairs You have pairs of DNA here. I hope you can see that. We have our pairs of DNA, right? That's what we made. We can synthetically make this, insert it into our genome or any other organism's genome. All right. I've done this in reality, constructed pieces of DNA and inserted them into other genomes. And so I'm going to take, we got to make a copy of this code. Well, this copy of the code is in this double-stranded thing. We're actually going to have to open this double strand up. We're going to have to unwind it and then pull the two strings apart. And then we're going to make a copy of one of the strands. Remember I said we're searching down one of the strands for code and the other strand has other code on it? One of those strands has the code we're interested in. How are we going to do that? How is the cell going to do that? How does it find this code and pull it out? And let's go back here. All right. So what I've done is I've just gone in and I've just pulled up a diagram that shows you a portion of the what's called transcription initiation. It's from my genetics class. And transcription initiation is, how do you start and initiate the transcription process? Transcription is taking that double-stranded DNA, opening it up and saying, this is where I need to copy some code out, pull it out so I have a strand. So what it does is you have a protein, and that is, I can't draw on here, but that protein is this TATA binding protein right here. And so you have something called a promoter. You see this thing that says core promoter down there, and you have the TATA box. The core promoter is a sequence that has TAATA, or something very similar to that sequence, has a bunch of T's and A's. And those bunch of T and A's come before where your coding region is. Right? So you have your coding region. That's the portion we really care about because it has the code that makes proteins we care about. But how do you know where those protein coding regions are? There has to be a promoter. There has to be something that tells again, another machine, a protein machine, where to find where you're going to need to start copying your sequence, because that sequence you need. Hey, I need to make some insulin. Well, you have a promoter in front of the gene for your insulin sequence. And when you need insulin, you're going to have to go into your genome, pull open your DNA, pull that piece of code out, make a copy of it, and then take it out to put it into your random access memory, and then take it over to your processor, and then make some insulin from it. All right, so how do you find it? In this really complex looking figure, but I'll boil it down to this. You have a protein. That protein can bind and looks for promoter boxes, all right? Promotion sequences. And I should have just kept this. This protein, what does it do? This protein is actually going around through typically what's called the major groove. So it's going around and around and around and around. It's bound to your DNA, and it's basically scanning down your DNA. And as it scans down your DNA, the TATA binding protein will bind and stick to the TATA box, and that's where the A's and T's are. And it can see into your inner core of your double helix. And it says, hey, there's a whole bunch of T's and A's right here in a particular sequence. In fact, it's more than just T's and A's. There's actually another sequence a little farther down that's like, let's see, I think it's TGA CAC. And so the TATA box is a certain number of bases, like 35 bases, that comes before the coding region. And the other little spot is somewhere else. So now when the TATA binding protein finds this spot and starts to bind to it, there's a bunch of other proteins, B, F, E, H, A, all these other binding proteins. All right. These are also they're called transcription factors because they factor into helping you transcribe. They bind to the TATA binding protein, and they do what's something called, they recruit the RNA polymerase. So this big red square or a rectangle, RNA polymerase, is a huge enzyme. All right, a huge protein. And RNA polymerase is the enzyme that's responsible for making RNA. It is the thing that copies your DNA and makes it into RNA. By the way, you also have DNA polymerase, which when you want to copy your DNA and make a new cell, you have to make copies of your DNA. You use DNA polymerase and you copy your DNA. In this case, you're going to use RNA polymerase and you're going to copy your DNA and make an RNA strand out of it. And so the TATA binding protein binds to the TATA box, and you can kind of think of it this way. It's kind of a weak point in the DNA because T's and A's don't bind to each other very well. They're much weaker than G's and C's. And so this is like a little weak joint in your DNA double helix. And so what will happen is once it binds there, the other transcription factors, the RNA polymerase, they're actually going to wedge open the DNA, the double DNA strand, and they're going to start to unwind the double DNA so that you're going to separate the two strands. And on the one strand that has the code that you're interested in, all right? Let's not get into that. We can go 20 different levels deep. On one of the strands, it's going to copy that code that's there into an RNA. And so the RNA will now have the code you want, and it comes out of the rest of the DNA. And then the DNA will just wind back up into that double helix, and everything will be just fine again, right? You don't play around with your DNA. You just open it up, make a copy, winds back up again. Now, the gist of all of this is to tell you that in order to even have the sequence available for your cells to be able to read it into a protein, you need to have some signals in the code itself that tell it, hey, guess what? You have a gene coming up, right? Your gene down here is like the sequence that's going to code for the protein, but you have promoters and regulatory sequences that come before the beginning of the gene, the coding region, that are signaling to the cell, hey, this is where you can find me. So let's go back and look at, we'll just submit this again. Let's go back here. Remember I said, all right, so this in our 5,000 base pairs of random sequence, we found an ATG and a TGA. TGA is also another stop codon in addition to TAA. So that sequence right there is readable into a protein if you can get that sequence out of your genomic code and brought out to the location where you need it. So you might be thinking, God, that sounds really hard. For one thing, it's going to require that not only do you have this sequence, but somewhere a little upstream, you must have a sequence with the promoter code. The promoter code has to be there too. Right? So you have to have two things happen in this random sequence in order for us to get an active, like actual protein being made. How are we going to determine whether that's possible from our random sequence? All right, so I'm going to go back here, and I can't remember if I have it copied. So I'm going to copy it again, and we're going to go to the Berkeley Drosophila Genome Project, because they have a little tool here that we can use. Even though we're talking about humans, fruit flies are going to work the same way. We've put our sequence into a eukaryote, an organism that has a nucleus. We're going to paste our sequence in here. Great. So there's our 5,000 base pairs. And what we're doing now, I didn't really say what we're doing yet. We're searching for promoters. We're searching for sets of sequence that the computer here can identify as this sequence right here has enough similarity to a promoter sequence, or exactly the same sequence, that if you were to make this sequence, these proteins that you naturally already have in your cells would recognize these sequences, and they would start to bind to it, and they would start to pull apart the sequence, right? Unravel your DNA, your double-stranded DNA, and it would open it up, and it would recruit the RNA polymerase, and you would actually start making a copy of some of your sequence at that point. All right, so we're going to ask ourselves, are there any promoters? Are there any promoters that we could predict are in our 5,000 base pairs? Let's do it. I hit submit. And okay. So on the forward strand of the 5,000 base pairs, we found 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 potential promoters, potential positions. Out of the 5,000 base pairs, there's 15 different locations where it appears there is a set of sequence that is similar enough to the known eukaryotic promoters. In other words, things we've identified in human beings, Drosophila, mouse, all kinds of vertebrates. We recognize that our genes all work the same way in terms of our biochemical machines. They're looking for the same types of places. And so just looking through is, oh yeah, here we go. I see TGCACT. I know that TGCACA is a perfect sequence code, and it's just upstream from, you see the bold letter here? It's saying that there is sequence, oh, here it is, TATAAA, all right, or ATATATAA. Yeah, TTTAAA. Almost all these have some sequence of T's and A's, so that's your sort of your weak joint. And then down here in a lot of these, there's probably going to be that ACACA thing going on. And over here, so this one's, so these have relatively high scores, meaning there's a high degree of chance that you're going to have enough sequence similarity that the enzyme, or the DNA binding proteins, the TATA box binding protein, is probably going to be able to connect to some little piece of DNA here. Now, this doesn't mean it's perfect. You don't actually have to have a perfect sequence. I'm sure that you have some genes in you that have promoters that have a mutation in them such that the binding site is not exactly right. The promoter site isn't perfectly right. And that can actually have an effect on your phenotype, on your function. Because let's say that you had a mutation in the promoter for making insulin. You wouldn't be able to bind as tightly to it, in which case you might not turn on the insulin gene as often. Right, so you're supposed to be making like 100 copies of it, and now you're only making 10 because this protein is kind of weak and it's falling off the DNA. So it's scanning your DNA in 1,000 cells, and only in 50 of the cells does it bind well enough that you get the operation going, and the machine only works on 50 of your cells instead of 1,000 of them. So overall, you're not making as much of that protein, but you are making some in some of the cells. All right, and maybe you're making enough, right, to survive. This is the way it is with a lot of genes. That it's not just that you make a specific amount, but you need to be within some range. And the range is often determined by differences in promoter sequences. There are lots of genetic diseases that are caused not by a problem with the actual gene sequence you have. You don't have the wrong protein. You can make the right protein. It's just you're not making enough of it because you don't have the right regulatory sequences and promoters. They have slight differences between individuals. And so the point here is that we found 15 on the first strand. It looks like we have like 13 or 14 on the reverse strand. So that'd be like 30 different promotion locations, which the computer predicts like, yes, your innate biochemical system, right? The machines I have inside of me would look at the sequence and find 30 different locations where it would be tempted to think, oh, I think there might be a gene here, and it might start trying to transcribe your sequence. And if it starts to transcribe it, it could transcribe for hundreds or thousands or even 10,000 base pairs downstream. If it transcribes for a couple thousand base pairs, essentially what's going to happen is almost all of your 5,000 base pairs that we inserted into your genome is going to be transcribed into RNA. That's biochemical activity. And that's kind of my main point, is that I hear all the time, oh, the ENCODE project shows us that like most of the genome has function. It has biochemical function. Like, okay. It has biochemical function. That means that what they could do is you can go in and pull out the RNA inside your cells, and you'll find out you'll have a couple copies of RNA from just any old random fragments of your genome. But having RNA, having biochemical activity, actually transcribing bunches of portions of your genome's being unwound and being transcribed in little pieces all the time, all over the place. But they don't necessarily do anything for you. Right? It's just the sloppiness of your biochemical activity that you happen to have codes that are similar enough to real genes that they're just making stuff, right? They're just doing activity. What happens? You ever wonder what happens to those RNAs? the stuff you transcribed, it just floats around. Eventually you have other enzymes that chew it up into pieces and you just recycle it and use it for something else, right? It doesn't actually become anything. Okay, so in our 5,000 base pairs, we found, I'm already forgetting now. We found, here's our ORFs, let's just do that again. We found a whole bunch of locations that just by chance have the right start codon, the right stop codon, and a readable sequence in between. We found 30 different locations where there is a promoter. And guess what? If there's a promoter, the promoter doesn't necessarily have to be right in front of the coding sequence. Let's go back over here. All of these different bold letters, I think I skipped this. All these bold letters is where transcription starts. So in this region, there is enough sequence similarity to a known promoter site in Drosophila, but it might as well be human beings, that it's very likely that this will entice those proteins to bind and recruit the RNA polymerase, and will start to do the whole operation of opening up the DNA. And where will it start to copy the DNA? You'll start copying it right here into RNA. And like I said, this could just keep going on and on and on and on and on and on and on for thousands and thousands of base pairs until it finds, by chance, in the case of our 5,000 base pair random sequence, it will find a termination sequence. Right? It might find it really soon, but it might not find it for a long time. And so if all 15 of these are places that will be just like trying to transcribe, and they're at different locations, well, we know what the locations are, at 4,788 to 4,838, and one's at 692 to 742, right? You have them scattered across your entire 5,000 base pairs. They're going to be opening up the DNA, unwinding it, and transcribing. And the transcription pieces are probably going to be running into each other. And eventually what's going to happen is I'm going to say, and I'm going to predict, based on what I'm seeing here, that if you were to insert this 5,000 base pairs of sequence into your genome, and then you were to look at your genome, and the way you look at it is you would extract the RNA from your cells and you were to sequence it, you would probably find 80% of the 5,000 base pairs in the form of RNA sequences that have been transcribed. Right? And so what's going on there? It's just what happens. It's just what cells do. It's just what biochemistry does inside of cells. Now, the really interesting question though is that biologically meaningful? Is it biologically meaningful information? Now, that's where I have to say it depends. I'll say that 99.99% of this, I'll call it randomly transcribed information, just because by chance there's going to be promoters, because the promoter sequences are very simple. And by the way, if you haven't already figured it out, why is most of your genome transcribed in small little bits all over the place? Because these promoters are, if I were to just take your genome and put it in this promoter finder, it's going to find a million locations where it could potentially copy your DNA or make RNA. All right? And it's going to be all types of locations that are not just your genes. So it's not shocking, not at all shocking, right, that we can identify biochemical activity coming from your DNA genome all over the place. Like lots and lots of your genome is being biochemically activated. But being biochemically activated in the sense that it's making little transcripts of it is a far cry from saying it's actually doing something in terms of like making the features of me, right? Running my operating systems. So what are they doing? As I said before, most of these are going to be transcribing a bunch of stuff that is only RNA. RNA can bind to other things like other RNAs. So if you have another RNA, and so this would be one possible negative impact of me splicing a piece of DNA into you. Let me go back here real quick. So this would be one of the negative aspects, potentially, of me splicing a 5,000 base pair sequence into your genome, is that out of that 5,000 base pairs, if 80% of it, 4,000 base pairs, do some quick math there. If you took 4,000 base pairs and you said, like, I'm going to make like, those 4,000 base pairs are in the form of like 10 different little strips of RNA. So you have A, U, C, U, 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 A, C, U, G, U, U, whatever, you know, the corresponding codes. And they're just floating around in your cells now. They potentially could bump into other RNAs that are real genes, right, before they're being read into proteins. And they might, if they have a similar sequence, make a double strand with them. And that's called RNA interference. And so RNA interference is where you, in some cases, you really do make an RNA on purpose. So one little piece of the DNA's responsibility is to make an RNA, and that RNA is made to intentionally interfere with another gene because maybe you don't want that gene to continue doing what it's doing. You're like, one way to block it is like, I'll make an RNA that purposely binds to that RNA and stops it from being read into a protein. The other place you use RNA interference is like, say you have a virus and you're like, I want to stop that virus from making copies, and they're an RNA virus. You could make an RNA that binds to that RNA of that virus and then potentially block its activity. And that, we would say, that has a function, right? Now there's an example where that little strip of sequence has a function. All right, so now the 5,000 base pairs of random sequence that we put into you, it's generating little bits of RNA, maybe a long strand of RNA of 1,000 base pairs, maybe little short pieces of RNA. And those RNAs can fold up into little structures. And maybe those structures might bind to something else. And if they do, probably they're going to interrupt the activity of whatever they're bumping into. And so if you have a really important gene and one of these random sequences happens to be very similar to that sequence or have the opposite sequence, so it binds really well to it, it could potentially change the function of your proteins, right? Or stop one of your proteins from being made, which then would have an impact on your function. I would say probably that's going to be a negative impact. However, even a negative impact is a function, right? And so you couldn't deny in that case, if it hurts you, you could not deny that that new random sequence that you just stuck into your body actually has a function, and it's actually changing the way you operate. And you'd have to say, I think, that it's new information, right? And so it's new information that is causing a change in the function of that organism. All right, another way that it could affect you is it might actually make proteins. I think out of that 5,000 base pairs, since we have 30 promoters, and we have, if you make long strips of RNA, and in that long strip of RNA, let's say you made 1,000 base pair RNA, like 20%, right, of it is all one long strand. We already know from our other predictor that there are 30 different, or what do we find, 56 different locations where there are short pieces that have a start and a stop codon. And so maybe on that long RNA, there might be multiple different locations where there are stops and starts. And your ribosomes could very well likely look at those and go like, hey, I'll start reading the sequence here, and I'll stop here, and I'll kick out this protein, and voila, you're making proteins. You're actually making proteins from random sequence. Now, what do those proteins do? Probably nothing. But again, they may interfere with some other operation, in which case they'd have a negative function. Most of them probably are inert, so we'd say they have a neutral function. They don't have any effect on the overall phenotype of the organism. A few of them, though, they might bind to something and change the function of something else. Or they may have their own function themselves. Now, one of the easiest ways to see that is that if they themselves bind to DNA or bind to another component of what's called a transcription factor, that will affect the transcription of another gene. And if it does that, it could upregulate it, it could downregulate it. It could turn on gene a little bit more, or it could turn it off. I know I have one more figure. It's the same figure, but I'm going to get a lot more out of it this time. I didn't show the rest of this. All right, so I was pointing out that the TATA binding protein and the RNA polymerase. Now here's the other thing about ENCODE. All right, it predicts that vast amounts of our genome are actually being transcribed. And for the reasons you've just seen now, there's lots of promoters. The promoters are really simple. The TATA box is a very simple thing. It's a very simple code, and you can find it all over the place. And so this process of binding to that spot, recruiting the RNA polymerase can all happen, and you can transcribe your DNA. Now, here comes another level of complexity. If all you have is a TATA box and what's called the core promoter, and you bind to it and you start to transcribe, you can transcribe a little bit, but it's not very efficient. So you might make a transcript, but other genes in your cell, and typically to have real physiological like activity and really make a difference, you sometimes might need 50,000 or 100,000 transcripts of something so that you can make a lot of something like insulin in a cell. So if you just spin off one transcript every couple hours, you're not really going to have much of a physiological effect. You're making a very, very small amount inside of it. That little strip is a tiny little thing inside of a sea of an entire cell that has billions of other molecules. And so it doesn't really have a lot of physiological effects on the cell because you don't make enough transcript. And so the ENCODE project can see even little tiny amounts of transcription being performed. And so it says like, yes, almost all of your DNA is transcribed, but most of it's not transcribed to the point where we would really care about it. It's not like you're wasting that much energy. It's not really bumping into too many things. Even if that transcript by accident can bind to a really important gene of yours, a really important protein, if you have 100,000 copies of that protein, you only have 2 copies of this thing that's binding to it, it's not going to change the way you feel. All right, you're still going to be doing what you're doing. You're not going to notice that 2 of your proteins were knocked out by this bad sequence. You're just not going to notice it. So what matters then? If you have a gene that you really need to use, you not only need to bind to the TATA box and recruit the RNA polymerase, you need to stimulate. You need to enhance, all right, that particular location and say like, this location is important. I need to make a lot of transcripts here because I need to express this particular gene. And so what a more important gene has, actually I'll say almost all genes actually have this. Almost all genes have other sequences that are upstream. All right, so here's the TATA box. Now if we go around here, we might go to 500 base pairs. And we find there's another sequence called an enhancer. You might have multiple copies of this enhancer. There are also sequences called silencers. Enhancers can attract a protein called an activator, right? And that activator, if you have that activator inside of your cell, right, that activator will bind to the enhancer. And the enhancer has a different sequence, and it binds to this activator, which has a different protein shape. And so it's looking for a different sequence as it scans down your DNA code. That activator then binds to what's called co-activators, which are other proteins in your cell. Those proteins bind to your TATA binding protein box, and they form a giant complex. It's called the mediator. I think I got that right, the mediator. And if you have multiple enhancer sites, you can attract multiple activators, and the multiple activators will attract more co-activators, which then drive up let's just call it this. They enhance how strongly bound your original DNA polymerase and TATA box are, and they promote or stimulate you to make more transcripts. And you can think of it this, they're going to hold that DNA open for a long time. Instead of just like, unwind it a little bit, make a little transcript, it sort of folds back up and the TATA box protein falls off because it's just weakly holding on to that TATAA. Now what you're doing is you're like grabbing onto it and you're saying like, you got to stay on there, right? I'm holding you in place. The DNA is wrapped around and the enhancers are there. They got all these other proteins. You've made this giant complex. It's a huge machine. And you're like, let's do it. Let's just open up this DNA and just let it rip, right? Let's let lots and lots and lots and lots and lots of transcription happen. And then we can like make a lot of this protein because we need a lot of it. Okay, so the random sequence you put into yourself probably, like, all right, first of all, it has to have coding regions to make proteins. Then the coding region has to have some kind of promoter somewhere upstream from it. We showed that, yes, there will be random promoters generated, but the enhancers are a little bit longer and they have different sequences. And they have to be somewhere near this whole thing. Right? And they have to be the kind of enhancer that's going to attract the right kind of activator. There's a bunch of different activators that have different enhancer sequences that interact with different genes. So it's going to be even less likely that you're going to have all those elements together. So out of your random sequence you placed into you, as I said before, I think that 80% of it is probably going to get transcribed, but it's going to get transcribed at a low level. A biologically unmeaningful level. Because even if it does bind to something else in your cell, it probably is not going to really affect you. You're not going to be like, I noticed that. Right? It's going to be such a small change in you that you're just not going to notice it. However, if there happens to be a couple enhancers by chance, happen to be near it, then you can enhance it. Well, then it would make more of a difference. All right, so back to the big picture. I feel like there's one other thing I was going to do here. Oh, let me just put this up for now. I forgot. I should have been pointing this out before. We've been talking about DNA, talking about you've got to transcribe it. And I'm saying it's pretty simple to show that even random sequence is going to get transcribed. And so again, going back to encode and how creationists talk about it, intelligent designers, and they're always like, oh, look, most of the genome is transcribed and we just don't really fully understand what its function is yet. Well, it's not surprising that it's transcribed. Being transcribed doesn't mean there is a function, right? It just doesn't, right? Showing that there's transcription does not mean that you eventually are going to find functions for all those transcribed things, right? And that's part of what this exercise is for. Imagine I'm not going to do it now because this has already taken too long, but if I went back and I've already done this, 100,000 base pairs, 100,000 random base pairs. And I were to take that sequence and insert it into a genome, how many would you get then? Well, I already know that I've got, out of 100,000, I had something like, 275 different protein coding sequences were in that 100,000 randomly generated sequences I came up with. And it had, I think it was 325 promoter sites, potential promoter sites. All right, so again, I think that 80% of that 100,000 base pairs I would insert into a genome, I would expect you could actually see that it has metabolic activity, that it is generating transcripts, and that in other words, my machinery or your machinery would be trying to read it. It would be like thinking, oh, there's information here. Let's see what we could do with this. They would be trying it out. What does this stuff do? Right? It wouldn't be completely inert. It wouldn't just be a random piece of sequence and you do nothing with it other than maybe copy it into another cell. Right, so transcription happens, and I am going to say that I think even out of the 5,000 base pairs, I would not be surprised if you could identify 4 or 5 proteins that would be generated from that random sequence that you would actually find in reality being expressed inside of your cells. They would be at low frequency, and they probably would be biologically unmeaningful. Now, the flip side of all this, not the flip side, the other consequence of all this stuff is that You might hear like genes from the junkyard. That's a phrase that's used for explaining how maybe genes can be derived from non-coding sequence, new genes or new genetic information. And I said before, if you inserted 5,000 base pairs of sequence, I had the feeling that most creationists would say, well, that's just random sequence. And so that's not biologically meaningful. And so it's not new information. It's just more chemistry. It's more molecules. Because no one can deny that there aren't circumstances where, viruses infect cells and they bring DNA with them and they can insert their DNA into the genome of the host. And so hosts gain more sequence over time. And certainly our genomes and all other organisms have bunches of extra pieces of DNA that have been added to them over time. And you can usually consider those extra sequences as, especially if they come from a virus or a plant or something like that, they're not going to be, obviously they don't have function for a human being. So they are just extra DNA. So I would say if you took a chunk of a plant, piece of a plant, and you were to say, like, I'm going to take 100,000 base pairs of sequence from a plant, and maybe it includes a couple genes in a plant, and I were to just like insert them into you and ask what happens, I would say you would still end up transcribing a whole bunch of them, and you would have a whole bunch of copies of RNAs that are sequenced that were originally from that plant that now you're making, and they would interact with inside of you with all of your different proteins, and they'd be at low levels. They probably wouldn't matter. And you might even make some of that protein that was made in the plant, right? Because they have similar promoter sequences as well, right? But you wouldn't necessarily be able to do anything with it. But I wouldn't, as Purdom and others, I've certainly heard many on, especially like Standing for Truth and a couple other YouTube channels, making a big deal about how there's all these transcripts and all like this metabolic activity, and you have like, these things being expressed, right? This sequence is all being expressed, right? Yeah, take a plant, put it in you, it'll get expressed. That is very different than saying like, oh yeah, that sequence is actually making you who you are. Like actually making functions that are meaningful for me biologically. Give me a second here to think of all the things I'm forgetting to say. There's so many little side trails one could take on this whole thing. And molecular genetics is really, really complicated. But I do think that what I've distilled it into in terms of the basic steps from DNA to RNA, the fact you need promoters, enhancers, and so forth in order to help a cell actually get activity out of things. It gives you a sense for what could happen with just random sequences. But what I've been talking about now is how you might now be able to envision how a new gene might be created, like a real new gene that actually does something, right? If you take 100,000 base pairs, let's just stick with 5,000. You take 5,000 base pairs and you stick it into something, or it gets transferred from one organism to another. And at first that organism, it can't really do anything with it. All right. None of those sequences mean anything. They're just random jumbles of either RNA, or some of them might actually become little pieces of proteins, but the proteins might not do anything. But then what could happen is that, if the organism moves into a new environment, and this is true for, I think this really fits the ice fish example. Fish that live in really, really cold environments, if they have portions of their sequence, which formerly were just random sequence, they weren't actually genes that were promoting and turning on and had like a really high function for, but they're just making sequence, right? All the time. They're just transcribing sections of their genome because they can't help it. Just like you and I are doing in all your cells right now. You're just copying little bits of pieces all over your genome, and the vast majority of them aren't going to do anything. But as they're doing that, let's say that this ice fish is living in a super cold environment, and one of the proteins one of the DNA sequences has a mutation. And that mutation now makes it fold a little different the next time it's made. And now it's making a protein that's folded a little bit differently. And that folding happens to act as a type of antifreeze. Having that particular protein in the cell helps the cell not freeze and make little ice crystals, which are damaging to the cell. All right, if that happens and that fish then survives better because this, what was a random sequence that was just generating a random protein, but now the protein is actually helping out and helping the organism survive, it has offspring, well, it's going to copy that mutation in the next generation. What if there's another mutation in that same gene that makes it, in some individuals, even a stronger antifreeze? Well, then they survive even better, right? So the organism is adapting to its environment, and the way it's adapting is it's having new mutations. Not that it's making the mutations, it's just randomly happening, but as they're happening, they just make a better protein. And then natural selection selects for, hey, out of all these different variants you just made, these work really well. Over time, what happens is that particular protein becomes better and better and better adapted. And then I just showed you the promoters. What if one of the promoters was originally only like a 90% efficient or 80% efficient? And then there's a mutation so that it's TATAAT instead of TATATT, which isn't as efficient. So the T changes to an A. All of a sudden, the binding is stronger, which then makes more protein. More protein gives the fish a better benefit. So those fish that have that mutation survive, have more offspring, copy that particular mutation, and then it has another mutation, maybe another sequence downstream changes one base pair, and now it has an enhancer. And the enhancer then increases the amount of transcript being made, which means the fish has even more antifreeze. You see, it's like baby steps. All along the way, the fish is okay. I mean, the fish survived before, but now it's surviving better. Or you could say the fish was surviving in somewhat warmer temperatures and couldn't really venture into the coldest temperatures. But then as it has mutations, it can venture into the colder temperatures. It turns out in the colder temperatures is where there's a lot of food resources that a lot of other fish can't handle, right? Can't access. So there's a huge benefit to being able to have more antifreeze and be able to survive in a colder environment. So natural selection is very strongly selecting for any new changes in this sequence that allow for more transcript to be made and more protein to be made. And so there are many examples of this in the literature now where we've shown that it's very likely that organisms had just random sequences between other genes that were doing real functions. And they were being expressed, though, just by this process of, well, metabolism. Biochemistry does this, right? It finds little promoters and it just spits out a few little copies. And usually that wouldn't matter, wouldn't do anything. But once in a while, because those little copies are there, they might find a use in a new environment. And once they do, natural selection is like, let's keep working with that, right? Let's continue to refine that and make that into a better protein. Through tweaking that particular, what was you would consider a random sequence, is no longer a random sequence. It's actually a sequence that you could say that sequence has a function. It's making a particular protein that has this particular function for the survival of that particular organism. That is not only not a far-fetched hypothesis, that's one for which we've actually seen the intermediate steps in several different examples, including the ice fish example, but also theoretically is highly likely now that we understand more about all these different promoters and the way molecular genetics works. All right, and so I hope that my little exercise here will help you see that even just randomly generated sequence our biological systems will look at random sequence and try to find a function for it. All right, and it's not trying like anthropomorphically thinking I want to have a function for this. It's simply that our biochemistry is set up in all living things to look for places where they can turn things on, express things, and then see what happens. See what will this thing do. Right? Now, how we control our genes is because obviously you don't want all your genes on all the time. Many of your genes you don't want on at points in time. And that's why, I guess I should show this One more time. Let's go back to this one more time. Did you notice that there's this other box up here called a silencer? Right? There are ways in which you can turn genes off. And there are other binding programs you can bind to the TATA box, which will then not allow the TATA binding protein to bind, in which case you can't turn on and transcribe or transcribe that particular region. And so that's one of the ways that we control, like, okay, I want to have this gene on, I don't want to have this gene on. You can many times turn genes on at a low level, but that doesn't really help you. So you have the enhancers, and if you have the activator around, and by the way, you make activators at other places in your genome. So if you want to activate a particular gene that has a particular enhancer, you can go make the activator. The activator then will go bind to the enhancer. The enhancer will then help bind to the TATA binding protein, which will then enhance this activity. Right? And then after you don't need something anymore, you can actually destroy the activators, and then you can lower the activity of a particular genetic locus position. Or you can bind something to the silencer, and the silencer can put a clamp on the whole thing and just say, like, forget it. I don't allow you to use this gene at all. Right? So those are all the complicated ways that things get they get controlled and regulated. Any brand new gene is not going to have that much control over it. But a new gene is not going to be like a centrally important, super like, hey, your body will only work if you have this gene functioning. No, all new genes that come into existence. There's a great study in rice that shows, like, between different species, there's like dozens and dozens and dozens of brand new genes that have arisen in very recent times. None of those genes are like critical function genes. They're all peripheral genes that that play very minor roles in tweaking the functions of the organisms and making the species be slightly different from one another. And that's the way new genes begin. They begin on the outside edges of metabolic networks, we'll call them, and they have like minor roles. Hey, I'm helping you to control this or that. I'm maybe turning something else up and down a little bit, right? And then over time, what could happen is they can become more and more important, right? As organisms that change in the environment, that particular gene might take on a more important role slightly over time. And pretty soon they may find themselves to be really important genes. But that might be a long time later. And we can look at other important genes today that are in organisms like all mammals. And you can look in the past and you can see like that particular gene wasn't necessarily a like supercritical gene at one time. It started out its life as a less critical gene, a minor sort of thing that like, hey, if you have it, you survive better. But if you don't have it, you can still survive. To finding itself more and more into the central core of the operating system of an organism. And once it's in the core of the operating system, it's like, then you don't play around with it. You're like, I'm super tightly controlled. By the way, if you look at different genes on a genome and you look at their regulatory control, the genes that are like really important have like really like complicated regulatory systems around them because you want to control them. Like you can't let super important genes get out of control. Otherwise, you won't survive. Other ones that are kind of like, whatever, you turn on a little bit, you turn on a lot. Oh, you're broken, you don't function all? That's all right. You can still survive without that particular gene. After all, you have hundreds of broken genes, but none of the important ones, right? Because if they were important, you'd be dead. And so natural selection is going to select for tight control of important genes, loose control of less important genes. Over time. So that's why you can take a 100,000 base pairs of random sequence and, the organisms are like, I can do this and that with it. And I'm like making little pieces and I'm making some proteins. And one or two of those might actually have a little tiny bit of function. But they're not going to be tightly controlled at first. They're like, if they function, they do. If they don't. But then maybe they have a few mutations. And like I said, if they become a little more important, natural selection will begin to say like, oh, stop playing with this thing. Let's start using this and putting more controls around it. We'll evolve regulatory networks to go with it. Lots of papers about evolution of regulatory networks. Lots of really interesting papers, like taking out a regulatory element and replacing it with random sequence, and then growing the organism for many, many, many generations. And what you find out is that after a while, the mutations that change the random sequence back into the sequence that is a better regulator, right? That's just direct proof that natural selection will select for greater regulation of a particular gene to enhance the activities of that particular gene. All right, so by taking the regulatory sequence out, they actually damage the organism in the sense that it doesn't survive as well. And so any new mutation that brings back the original sequence will then be natural selection. We're like, great, I'm glad we're returning back to the original sequence because that's where you were best fit for the environment. But still, the mutations were random, and you're taking random sequence, and it can be turned into, by natural selection and mutations, back into the original promoter sequence that it was. It's very predictable. And that's what you see with all these. I just showed you that whole list of promoter regions. They all kind of looked a little different because they have different sequences, but they all have some similar patterns. And if those patterns lead to making proteins that end up having functions, you can bet the patterns will become more and more and more and more and more similar, because mutations will eventually make them into ta-ta boxes, all right? And the other, I think it's the cat box, all right? The TGCA, I can't remember now, GCTACAC. Eventually they will coalesce onto very similar sequences to all other mammals that have those. All right, now I am just rambling because I can't remember all the other things I was going to say. But I hope you got something out of this exercise. I'd like to do a lot more bioinformatics stuff like this. So you only saw just a tiny little bit here, but I'm kind of building up to doing some sequence comparisons and then actually showing, like, looking at sequence comparisons and maybe making like little phylogenies for mitochondrial DNA, or looking at Y chromosomes or something like that to test things that people say all the time, as if like just saying it makes it true. It really helps to actually see the sequences in action and look at actual sequences of organisms and to think about how the sequences came to be the way that they are. All right, let's call it quits there. My name's Joel Duff, and you're watching my channel. And if you enjoyed this content, hey, subscribe, hit like, all that good stuff. We'll talk to you later. Bye-bye.